Hi, and welcome to Microbiology. This is our Chapter 1 lecture from the Openstax textbook titled An Invisible World. This will be a broad introduction to microbiology. This chapter is divided into 3 parts. Part 1, what our ancestors knew about microbes or microorganisms. Part 2 is a systematic approach which looks at the naming and organization of microorganisms. And Part 3, an overview of the different types of microbes that we know of so far. So microbes are ubiquitous. That means found everywhere. And they can be found in Yellowstone, like these hot springs on the left, all the way to underneath the Antarctic ice, like this shown on the right. Most of them are not, we can't see them with the naked eye. So they're called microorganisms or microbes because we need microscopes to be able to see them. And even though a lot of them have a bad reputation, because we don't usually hear about them unless they're in the news for causing food poisoning or some kind of recall of maybe like Caesar salad, we actually have found that most of them are harmless. And in fact, we need them in our daily lives. They benefit us, like the microbes in our gut or the microbes in our skin that protect us from the more harmful microorganisms. And of the beneficial microbes, these include those that can help us make biofuels, such as algae and others. We even have microbes that help us make medicines, like penicillin. That antibiotic is produced by a fungus. And many of the foods we enjoy are produced with the help of microbes, such as adding different types of bacteria or fungi to ferment and produce cheese. We've got beer here. And yeast. If you make bread at home, we add yeast to help us make our bread rise. There are trillions of microbes on and inside of our bodies. Some of them are critical to our health, and others can be pathogenic or able to cause disease. Even though we know a lot about many microbes, there are so many more that we don't quite understand very well yet, as well as many more that we probably have not discovered or encountered yet. So there's constantly new research and new technology being developed to better understand the currently unexplored. Our book has another example of beneficial microbes, including the bacterium Alcanivorax borcumensis, which is a naturally occurring oil-eating marine bacterium, but can also be added to a spill if cleanup is needed. And as we go through each chapter, keep these learning objectives in mind. You'll want to be able to answer the objectives at the end of each section in order to not only prepare for future quizzes or exams, but also make sure you have an understanding of the main points for each section. Section 1.1 begins with a clinical focus problem, which if you haven't read it yet, I recommend pausing, reading it, and coming back to the video. In summary, a 41-year-old female has been experiencing headaches, a high fever, and stiff neck, goes to the doctor who suspects that she might have meningitis, and meningitis can be caused by microbes such as bacteria, fungi, viruses, or it could be non-microbial, so it could be an outside cause, another reason. But the doctor takes a cerebral spinal fluid or CSF sample by doing a lumbar puncture around the spinal cord, and these samples will be sent to different labs for testing. If it's microbial, the CSF is usually probably going to be discolored or be cloudy, whereas a normal CSF is usually clear. And one of the first questions our textbook asks is, what types of microorganisms would be killed by antibiotic treatment? What do you guys think? We know about microbes or germs today because we have the tools and technology to see or detect them. And these include things like bacteria, viruses, fungi, and a general term parasites that we'll describe more later. But before microscopes were invented, it was pretty much impossible to prove that these microbes existed. I mean, it's hard to prove something exists if nobody can see it, right? So despite this, people knew something useful is present, right? People have been using microbes for thousands of years to make fermented foods or even prevent or treat disease, for example, through herbal medicine. But this means that, even though we knew something was there, we didn't really understand these microbes scientifically. We didn't really know what was actually happening. And that is the main goal of this section. So fermented foods like bread, cheese, yogurt, beer, wine have been enjoyed for thousands of years. So we know that people had some idea of microbial life since they were able to take that knowledge to make these foods. So fermentation in general is a process that uses microbes such as bacteria, yeast, or molds, which is a type of fungus, to convert sugars into alcohol, gases, and acids. And we're going to see this in more detail in our biochemistry component. But early humans probably discovered fermentation by accident and somehow refined their processes to make these foods over time. And it became a key method to preserve food and make it taste better. Here's a picture of yeast that allows bread to rise. If you put it into a liquid mixture, you might see bubbles form over time. And we'll be looking at these individual yeast cells, which are a type of fungus, in lab soon too. This slide kind of makes me hungry, but these are some examples of fermentation products you might have at home. So maybe go take a look at your fridge, take a look at some of these products if you have them, and look at the names of the microbes that are in them that were used to produce these. So you might have sourdough bread at home, maybe kimchi, miso, kombucha. Take a look. Prehistoric humans had a very limited understanding of what caused disease. So different cultures developed different belief systems and explanations, such as blaming illnesses on gods or fate. But there is evidence that suggests prehistoric people did still at least attempt to treat these illnesses and infections. And one very famous example is Ötzi the Iceman, who was found in the Alps in the early 90s, and it was determined his body was over 5,000 years old. It was extremely well preserved because of that cold temperature. Evidence found that he was infected with different parasites such as Trichuris, Trichiura, as well as Borrelia burgdorferi that causes Lyme disease. And scientists think that Ötzi may have been trying to treat his own infections because of some things that were found with him or on his body. For example, they found this mushroom, Fomitopsis, on him, and he might have been using it because it has laxative properties to maybe help flush out his system, and also antibiotic properties, maybe to get rid of that Borrelia bacterium. He also had these tattoos on his skin that appeared to be or to have been made by cutting incisions in his skin and filling them with different herbs and then burning them. So maybe that was some kind of early medicinal treatment. So this is just one example of evidence that humans have probably been trying to treat illness using these different natural remedies that might have scientific evidence behind them, as we now know today, but using these early or primitive techniques. And this is optional, but if you're interested in seeing an actual picture of his preserved remains, you can click on the link that I've provided in the PDF version of this slideshow. And also there's even a movie, a fictional account of what they believed Ötzi's life was like. Several ancient civilizations seem to have some kind of understanding that disease could be spread by things that they couldn't see. And this is evident in different types of historical attempts to contain or stop the spread of disease, such as the practice of quarantining people. So quarantining often came too late, however, especially for diseases that were slow to develop, such as leprosy. So in leprosy, signs and symptoms, for example, might not show up for a year or 20 years later even. That means people were probably quarantined after they had already spread the disease to others. But ancient civilizations seem to recognize that clean water and sanitation were important for a healthy society, because we have several examples of evidence of this. For example, the Indus Valley cities built several different types of advanced sanitation systems, their wells, baths, and drainage. And then the Romans also had their aqueducts, as well as the Cloaca Maxima sewer system you'll see a picture of in the book. Even the ancient Greeks, there's evidence that they understood something that could be in the air to spread disease, and they called that miasma. So these early structures and infrastructures probably helped reduce waterborne diseases and outbreaks within these civilizations. Here's a picture of that Cloaca Maxima. This is also known as the greatest sewer built by the Romans. You can see down here on the bottom left, these are essentially their toilets. You can see a person would sit right there, and you could have multiple people going within that same unit. Do your thing. So waste would go through these holes and it would be draining downwards, carrying waste away from the city into the river. Several scholars also played important roles in helping us understand back then the invisible forces. Now we know they're microbes that caused infection and disease. And these include Hippocrates. He was a Greek physician, a doctor, also known as the father of Western medicine, that rejected supernatural causes of disease. Instead, he believed that diseases came from within the patients or from their environments. He's also credited with the Hippocratic Oath, which is an oath that doctors take to treat patients without harm. Here we also have Thucydides. He was a philosopher and historian who survived the Athenian plague, and he saw that people who survived the plague did not get reinfected again with that same disease. And this is an early idea of immunity. So he advocated for evidence-based analysis of disease. Finally, we have our writer here, Varro, who proposed that there was something in the air or water he called invisible creatures that could cause disease. And this is one of the earliest written ideas linked to microbial theory, which is the idea that microbes can cause disease, or diseases are caused by specific microbes. And those three are shown here. On the right is Varro again, and he is the one that noted that there were tiny creatures that could float in the air and enter the body through the mouth and nose to cause serious illnesses. We have two additional scholars in medicine here, including Al-Razi, who conducted experiments to test his theory, so kind of like the early scientific method. It's really interesting because when they were looking to build a new hospital, what he did was he took raw meat and he hung it at different locations within the city. And ultimately, they decided to build the hospital where it took the meat the longest period of time to go rot. So I think what they were thinking of is that location probably has the fewest invisible creatures, what we now know as microbes, in the air in that area. He was also the first to distinguish measles and smallpox, which we now know today are caused by different viruses. And overall, he emphasized the understanding of causes of illness, not just treating the symptoms afterwards. We also have Ibn Sina, who wrote the Canon of Medicine, a foundational medical text that's been translated into many different languages. And in his book, he had descriptions of contagion, breath-based transmission, quarantine, there was anatomy, treatments, and a lot of different types of substances they were using as medicine back then. So like those objectives, the check your understanding questions or examples are also important to kind of gauge your knowledge. Are you understanding what's happening so far? The first one shown here, give 2 examples of foods that have been historically produced by humans with the aid of microbes. So these include things we've talked about like bread, cheese, what else do we talk about? Beer, yogurt, sauerkraut. For the second one, explain how historical understandings of disease contributed to attempts to treat and contain disease. We just saw several examples, such as quarantining. We have sanitation systems like that Cloaca Maxima. We have early scientific observations like Thucydides, who found that people who survived the plague didn't get reinfected. Also Varro, who said that there were probably invisible creatures or tiny creatures in the air that could cause disease. So go back and see if you can answer these. Take your notes together, summarize them, and answer these components in addition to those objectives as well. So we can see that people in the past probably suspected that there were some kind of invisible creatures causing disease. But it wasn't until the microscope was invented that they could actually prove that they could see these microbes. And that's what we're going to look at next. The first microscope is usually credited to Antony van Leeuwenhoek, and he was a Dutch clothman who was also really good at grinding glass. Ultimately, he started building lenses and apparently the first microscope. He, it said, was a very curious guy, so he looked at everything he could get his hands on to look at under the microscope. And these included things like pond water, human samples, and within these samples, he saw things swimming around. These things he called animalcules, or wee little beasties, what we now know are microbes or microorganisms. So these are probably things like bacteria or protists he saw swimming in the water. It's also said that he was a really secretive guy, so he wouldn't let anybody else look under his microscopes. So nobody else was able to see what he could see, not even his wife. And he would also not be willing to teach others how to make microscopes. So it's said that the next microscope was not built by another person until around 200 years later. So here's a depiction of Leeuwenhoek on the top left. Here's his first microscope, and this is where, at that tip of that needle, where you would put your specimen and look through that lens to see it close up. Under the pond water or rainwater samples he saw were probably these different types of protists. So about 200 years after van Leeuwenhoek's microscope, we have this period of time known as the Golden Age of Microbiology, from 1857 to 1914. And this is marked by rapid progress in understanding microbes, largely due to discoveries such as those made by these two microbiologists, Louis Pasteur and Robert Koch. Pasteur is famous for many things, but some of these include proving microbes are what cause fermentation to happen, inventing pasteurization to prevent spoilage. If you've ever had pasteurized orange juice versus unpasteurized orange juice, you'll see that the unpasteurized orange juice boils a lot more quickly because it has not been heated to kill these excess microbes. Pasteur is also known for development of certain vaccines, such as the vaccine for rabies. And Robert Koch down here is famous for something that we'll learn more about later, that's known as etiology, which is the discovery that there are connections, specific connections between single isolated bacteria or other microbes and a known human disease. For example, he discovered that a specific bacterium called Bacillus anthracis causes anthrax. A specific bacterium called Vibrio cholera causes cholera. And Mycobacterium tuberculosis is the bacterium that causes tuberculosis. So very specific microbes cause these specific diseases, and that is known as the germ theory of disease. This is different from microbial theory, which is more general, and that latter one, microbial theory, really just tells us that microbes in general have the ability to cause disease. So these findings really helped lay the foundation for what we have today, what we know today in modern biology and medicine. And through all of this knowledge is really gained through scientific method or microbial research. Here they are, Pasteur and Koch. So we can see that proving microbes exist through van Leeuwenhoek's microscope and the subsequent experiments conducted by Pasteur and Koch really helped us better understand disease. So van Leeuwenhoek's microscope showed us the microbes and confirmed their existence. Pasteur showed that microbes are important in terms of fermenting our foods, but also spoiling our foods, leading to pasteurization. He also discovered those vaccines. And Robert Koch, we saw that he helped us understand the links between certain microbes and diseases. And all of this was really accomplished through a scientific method, right? Scientific evidence-based approaches, which really serves as the groundwork for what we have in microbiology today. Towards the end of section 1.1, you see this section in the book called the Microbiology Toolbox, where they have a list of terms, and we're going to see these items in our lab. It'll be a lot of fun. We'll have our microscopes, our stains and dyes. Eventually, we'll work with petri dishes and more media, test tubes. But I wanted to point out a couple things. The first being our inoculation loop. So this, the one on the bottom, you'll see has a little circle at the end. The inoculation loop is what we use to pick up bacteria and transfer it to another medium. The inoculation needle is another tool that we can use to pick up bacteria. The difference between the two is really the amount that you want to pick up. If you want to make sure you're picking up just a very tiny amount, you use the needle. If we want more, we use the loop. And the loop, I would say, is more commonly used. We're going to be working with Bunsen burners every day, and we don't have these. These are on my wish list to buy when we get more money for our department. These are micro incinerators to sterilize the loop or needle as well. But right now we're going to be using the Bunsen burners. Here's a petri dish shown on the left from our textbook, and we're going to be learning how to streak bacteria in such a way in the future, and another look at the inoculation loop. As I mentioned earlier, try to go back and see if you can answer these objectives. Look at how our ancestors improved food. We learned about fermentation to preserve foods and enhance their flavor. We looked at, causes of sickness and disease, how they're explained. So we looked at how originally people thought maybe disease was caused by gods or fate or different types of spiritual forces. But they also had an idea that sanitation was needed, such as through the cloaca maxima, right? Different sanitation systems. And then look at the different historical events associated with what we know today, that birth of microbiology, including Van Leeuwenhoek's microscopes and Pasteur and Koch's discoveries. All right, that's the end of part 1. In part 2, we'll look at the naming and organization of microbes.